Hello everyone and welcome to today's live broadcast, Modification of Glycans and Bioprocessing, presented by Ryan Boniface, a cell biology scientist level 3 at Bioproduction Research and Development, Thermo Fisher Scientific. I'm Susie Valdez and I will be your moderator for this educational webcast presented by LabRoots and sponsored by Gibco, part of Thermo Fisher Scientific. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on the green Q&A button located at the lower left of your presentation window. Type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing this presentation via a slide window. To enlarge that window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right or use the Q&A button to let us know that you're having problems. Please join me now in welcoming this scientist, Ryan Boniface. I will now turn the presentation over to him. Welcome, sir.
Thanks, Susie. Hello, everybody. I guess depending on where you are in the world, good morning, good evening, good afternoon, good night. So I'm going to speak a little bit about modification of glycans in bioprocessing. And I guess I'll get started by saying that I've been a scientist now for 10 years with Thermo Fisher, and I think it's pretty interesting the way our industry has advanced over the last 10 years. When I started here, there was a lot of times where we were performing cell culture experiments, and our goal was to get as much growth as possible. We were working with cell lines that didn't have necessarily a lot of productivity. And then over time, we started shifting our focus to try to get as much out of the cells as possible. So we started turning towards looking at specific productivity. How much protein could we get these cells to make? So really, we went from having less than a gram per liter, so now we've got anywhere from 2 grams per liter all the way maybe up to 10 grams per liter. So quite an advancement. And I would say over the last 4 or 5 years, the focus has started to shift now where the protein's there, the cells are there, but the quality maybe isn't. And we're really starting to really dive into that quality aspect of it because it's really important. And we really all need to kind of think about the quality that we're producing from our proteins. And, I tend to think of it, and I always have for a long time, that, I have customers that I have to develop media for and feeds and supplements so that I can help them with quality. So I tend to think of customers as anybody out there that's producing drugs and proteins and immunoglobulins. But the fact is we're kind of all really customers, right? I mean, there's a chance that there are going to be drugs that are being made that you or I might be using. Our loved ones, our family members, parents, children, grandparents. So really, we really kind of need to have a great deal of vested interest into this. We need to make sure that we're producing high-quality products because ultimately we're all going to be customers. We're all going to be affected by this. And I've kind of said multiple times when talking about this is that, it's one thing if you can produce the protein, but it's not going to matter if you can't get the quality. If it's not where it needs to be, if you're not targeting the right thing. And if it's not right, it could have detrimental effects to anybody taking the drug. Certainly it could elicit immune reactions and things like that, or maybe not be as effective as it could be. So really, as somebody that works for Thermo Fisher Scientific, we're incredibly focused on this. We're very passionate about making sure that our customers and everybody can have the protein quality that they want, that they're targeting. And this really fits with our mission statement, which is to enable our customers to make the world healthier, cleaner, and safer. And, going through this, we know that therapeutic protein quality is really important to everybody. It's important to all of our customers. And we also know that, everybody's looking to reduce costs and increase productivity. And again, like I've stated, glycosylation, protein quality significantly impacts the quality, the consistency, the efficacy of anything that you're producing. So really our challenge is to be able to identify these glycans, measure them, and really control glycoproteins. So I'm not going to get incredibly in-depth with glycosylation, but you can see here on this slide number 3, that these are some of the kind of more typical glycoforms that we're looking at. We've got terminal galactose, terminal sialation, fucose, manostructures. And these are all important for the effects that they can have on the drug. You can see in the case of galactose, increasing that would increase the CDC immune response. Increasing terminal GLICNAC, you can have longer serum half-lives, increase EDCC. So all these things are really important. Now, certainly this is just a very small subset of glycans that you could potentially be looking at. There's thousands of different combinations. But at least for a lot of our industry, we're kind of focused on some of the ones that I'm showing here. Certainly G0, G1, G2, and the fucosylated versions of that, as well as having a silated glycoform. So there's plenty of applications for glycosylation. Not every customer is going to be the same. Certainly everybody's going to be looking at different things. And we're going to have customers that are certainly, they've got a cell line and maybe they're going to be transitioning to a new clone or a different cell line. And they need to match the glycosylation. So that's something that there's a particular customer set that we'd be looking to help. Another one would be biosimilars. So a biosimilar is going to be producing a drug, and they have to match the glycan profile of an innovator molecule. So there's something where it's really important to target a very specific glycan profile. And then you can have another category where you can, improving the efficacy of a molecule. So this is getting the best glycosylation profile you can to increase stability, improve the pharmacokinetics and dynamics of a molecule. And this goes for innovators who are looking to have the best drug possible. But then another category, which we don't talk about all that often, are like the biosimilars, the biobetters. So these companies that are looking to produce maybe a generic drug, a biosimilar, but they know that they can improve the product quality enough where it is technically, a new drug, but it is vastly improved kinetics. So those are just some of the applications that we might be looking into. So I'm going to turn and kind of talk about glycans and their profiles in cell culture. So when we're looking at cell culture, there's a number of aspects that go into controlling glycosylation or what can affect it. And I've always kind of looked at this as kind of having two categories, one being the cell culture process and the other one being like your media components, your supplements, your feeds. But really it's a little bit more complicated than that. Certainly you have your cell line, which is one aspect. Different clones, different cell lines, whether you have a CHO cell line or maybe a 293 cell, they're going to have very different glycan profiles. So that's one aspect, kind of choosing a clone. The other one would be kind of what system you're using to grow your cells to produce protein. There are going to be plenty of differences between flasks, bioreactors, perfusion setups. And then, you do have your process parameters, your things like your pH, your DO, your CO2, temperature, mixing rates, shear. Those things will certainly have an effect on glycosylation. And again, that other category would be your media, your supplements. What kind of components you're giving to your cells. What kind of building blocks they have to build these glycan patterns or these structures. And when looking at this, we know that DO is important. If you can maintain your DO between, let's say, 10 and 100%, you can kind of ensure that you're going to get consistent glycosylation. Certainly, if you increase your CO2, there's a chance that you can decrease sialation. Lower temperatures can decrease sialic acid, but at the same time, performing temp shifts can result in maybe maintaining glycosylation, along with probably increasing and or maintaining specific productivity. And I spoke a little bit about differences between different types of cell culture. When you're comparing perfusion to fed batch cultures, there is a chance that if you're using a perfusion culture, you could be increasing your sialation on your protein. And then also something like your metabolites. In the case of ammonia, that can be very preventative to getting terminal glycosylation. Most of the things that I'm showing here on this slide, these are all available or widely available in literature. And to kind of show some examples of this, here on this slide is demonstrating what can happen if you do have a sort of increased ammonia concentration. And so what we have here is we have on the left, condition number 1, you can see there's a high peak for G0. It's probably about 87%. In that case, you look on the bottom, the ammonia concentration on that day, day 14, was about 9 millimolar. If you look all the way over at the right, condition 3, again, day 14, but this time the ammonia concentration is half, 4.5 millimolar. And you can see that there's a decrease in G0F by about maybe 10 to 12%. And you also have an increase in G1F and maybe a slight increase in G2F. And this would be pretty important for customers on harvest and maintaining the level of ammonia that's present in your culture. So obviously having higher galactosylation, G1F, G2F is important. And obviously minimizing the amount of ammonia buildup in your culture is also very important. As another example, harvest viability. I know that more often than not we want to extend our cultures as long as possible to get the most out of the cells, but it's also important to harvest the cells at a point where they're sort of in a good viability range. So on the left you can see there's a day 14 harvest, and the cells are at 95% viability. And again, G0F, the peak is about 70, 72%, and about 20% G1F. And if you look on the right, day 19, now we've got a 45% viability. You can see that G0 has gone up, and terminal galactosylation, G1F, G2F, has gone down. So this is a situation where this wouldn't be that very ideal. At the same time as your cells are dying and your viability is decreasing, you also have a lot of cell lysis and death, which is going to release plenty of proteases that can also degrade your protein and your glycans. So early on, when we were looking to develop a Glycan tuning product, I screened handfuls of components that through literature or other means, should have some impact on glycosylation. And when looking through these components, I found a handful that were functional. So if we look at this graph here to the left, and if you look at the very left, most peak, listed as efficient feed C plus control, you can see there's a very high level of G0F, almost 90%. And subsequently, you have very small percentages of G1F and G2F. And then if you look all the way over to the right, it's listed as 1X. You can see that there's a drastic decrease in G0F. So in that case, I found a handful of components that I know worked. And my first thought process is, let's add them to the media. Let's see how it changes the culture. So I added them in at what I deemed as a 1X concentration. And luckily enough, everything worked out, and I was able to get a sharp decrease in G0F. I went down from about 90% all the way down to almost 45%. And you can see that G1F increased quite a bit, and G2F increased quite a bit. So these components are working. That's great. This is exactly what I was looking for. So knowing that I can put them in at a high concentration, knowing that that works, my next thought process is, well, that's great for somebody that's looking to just maximize the amount of glycosylation that they have. But what about those biosimilars that are out there that are looking to target a very specific profile? Or the customers that are changing between cell lines and they need to hit an exact glycan profile. So in that case, knowing that these components work, my first thought process was, let's titrate them back into the culture. Let's add them in there at different concentrations and see if we can get predictable glycan profiles. And as I did this, you can see that if we move from just on the left, the efficient feed C plus control, to the very small amount of 0.03x, there was about a 30% decrease in G0F. And then as you move further along, you can see that there were very small decreases in G0F, maybe 5, at most 10%. And although this looks all right, it wasn't a linear response. Okay? And you can see just by adding that 0.03x concentration, that's a pretty small margin of error. And what we were finding was that there really wasn't a lot of consistency in this effect from experiment to experiment. And when I'm looking to develop a product, I have to develop something that's going to be easy enough for our customers to use. It doesn't do any good for me to release a product that I hand over, and when I get the question, how do I use this, how much should I add? In this case, it's kind of, I don't know. I can't tell you exactly how much to add, and it kind of introduces a trial and error scenario. So you might perform an experiment, you might titrate these components in and say, okay, it's maybe around 0.36x, but it's not what you need to be. So then you have to have a second experiment. And maybe that second experiment doesn't get you to where you need. So then you have a third experiment and so on. So really, I'm looking to reduce the amount of experiments that anybody would have to perform to get to target a specific glycan profile. It doesn't do me any good, and it doesn't do our customers any good. So this is a product that isn't easy to use, that requires multiple trials and a lot of effort and a lot of time and money on these experiments. So what we did is we abandoned that approach. Just titrating in those components just wasn't working. So what we came up with is this, what we're calling transition points. I'm going to kind of walk through this slide so that it makes sense, and I'm going to show an example on the next slide using this exact same table. So we have a series of efficient feed plus products. There's an A plus, a B plus, and a C plus. At the same time, we developed the Glycan Tune plus products. There's an A plus and a B plus and a C plus. And each one of those A pluses and B pluses and C pluses match up to each other from an efficient feed perspective and a Glycan Tune perspective. So let's say for this example here, we'll be using efficient feed C plus and Glycan Tune C plus. And this experiment, what we have is daily feeding from days 4 through 15. And the very top condition, where we just see it says efficient feed plus, what we would have is daily feeding with just the efficient feed plus product. For this example, we'll just say efficient feed C plus. So every day from day 4 through 15 was feeding just efficient feed C plus. And then if we look further down here on the very bottom, we have the Glycan Tune plus condition. So in this case, we're feeding the cultures a separate condition with nothing but Glycan Tune C plus from days 4 through 15. And then what we have in the middle, we're deeming as transition points. So let's take day 11, that condition for instance. What you would do is you would start feeding efficient feed C plus on day 4 and feed that all the way up to day 10. And then on day 11, you completely switch to Glycan Tune C plus. And from day 11 through day 15, you're only feeding Glycan Tune C plus. And obviously, the other conditions here are just moving that transition point around. So you're just changing the day that you're switching from the efficient feed product to the Glycan Tune product. So in the case of day 9, instead of switching over on day 11, you're just switching over on day 9. To show an example of what this looks like with efficient feed C plus and Glycan Tune C plus, here we have some data. And what you can see on the very left, again like that first experiment, we have just efficient feed C plus control. And on the very right, you have just the Glycan Tune C plus control. And I should state, which I didn't mention before, Glycan Tune C plus is a complete feed. So it's everything that you need to add back to your culture to keep your cells going with the addition of glycosylation enhancing components. And now what you can see is that we get a much more linear response by doing these transition points rather than the previous approach where we were titrating in components. So now you have this stepwise approach where you get more predictable and linear responses in your glycan profiles. And for this experiment, I was only showing odd days, but certainly, like if we take day 15 and day 13, those transition points, you can see that you've got maybe 70% G0F for day 15 and maybe 65% for day 13. But if you were to transition on day 14, you could more than likely have a result that maybe fell in somewhere in between that, so maybe about 67, 68%. So there's numerous ways to do this, and I'm sure that you could take this even further, and you could have this timed by the hour to get a very specific profile. So what this really eliminated was a lot of the guessing work with volumes through scale-up and other things like that, where now you're just picking a day to transition to the Glycan Tune feed. So this next slide, the graph up in the top right is the data that I just showed you. And on the left, this is to show that we can modify these glycans using this transition approach without really negative affecting the growth or productivity of the cell line. I think we had variation somewhere within about 10%. So this is an example on this slide using efficient feed C plus with Glycan Tune C plus. On this next slide, here is data for using Efficient Feed B+ with Glycan Tune B+. And again, on the top right, you can see that stepwise pattern. Same transition days as Efficient Feed C+ and Glycan Tune C+. You get a nice linear response and a predictable response. And on the left, with the exception of that one data point, which has a fairly large error bar, you can see that both growth on the top, viable cell density, and the bottom, IgG titer, very similar results, even though we have dramatic changes in the glycan profile. And on this next slide, here's an example using the Efficient Feed A+ product with Glycan Tune A+. Again, the top right, you can see the effects on the glycan profiles using these transitions. And the left, again, top graph, you have the viable cell densities, and on the bottom, IgG titers. So there's going to be plenty of instances where a customer or anybody for that matter they're going to have maybe a media and feed combination that they really like, but they're not getting the glycan profiles or the performance that they really want. And so they might want to try the Glycan Tune products with their current media and feed setup, and that's okay. In this experiment, we're using a commercially available feed with a commercially available media that is corresponding to that media. And we're using this particular media and feed to the manufacturer's specified feeding profiles. And what we have here is, again, transition points. So for this particular feed, the feeding days were feeding on day 5, day 7, day 9, and day 11. So what you have on the very left side, the left graph, or the left set of bars, with G0 about 75%, that's just this commercially available media and feed with no Glycan Tune. And then on the very right is the commercial media fed with Glycan Tune on days 5, 7, 9, and 11. So you can see a decrease in G0F from, let's say, 75% down to, looks like maybe 47, 48%. And then the other days, we did transition points again. So day 7, on day 5, we fed with the commercially available feed. And then on day 7, 9, and 11, we fed Glycan Tune C+. For the next set of bars, we fed day 9 and day 11 with Glycan Tune. And the last set, we fed just day 11. So days 5, 7, and 9 were fed with the commercial feed, and we only fed with Glycan Tune on day 11. So you can see that you might have a feeding profile, you might have another media, you might have another feed, but it's still possible to use these Glycan Tune products with your setup. And you can still do those transition points, even with feeding that isn't daily feeding, something that's bolus feeding every other day. And to really show the importance and the power behind these transition points in the Glycan Tune products, what I have here on the slide, and this really illustrates just how well this can actually work. If you look, there are 4 commercially relevant biotherapeutic glycan profiles that we've measured internally on this graph. And using the data from one of those previous experiments using Efficient Feed C+ and Glycan Tune C+, with those transition points that I talked about, we were able to pretty closely match the glycan profiles of each one of these commercially relevant biotherapeutic profiles. You can see that we're only within maybe a couple % of the target profile. So this is really critical for those biosimilar customers out there. They know that they've got to hit a profile. They're going to try to hit it exactly dead on as much as possible. I'm sure that there's some wiggle room, but they're going to try to hit it with 100% accuracy. So this is a chance for them to get really close with a very simple protocol. And in this case, to match these profiles, we had transitions on day 9, day 15, again, day 15, and then day 11. So another important aspect of these products, both the EfficientFeed Plus products and the Glycan Tune Plus products, is that they utilize our AGT technology. So because of this, they're really easy to reconstitute. It is as simple as just adding to water. They auto pH. There's no unnecessary steps to pHing, no long mixing steps. You literally add it to your water, you mix it, you bring it up to a final volume, you filter it, and you're ready to go. And at the same time, these feeds are super concentratable. So for the EfficientFeed products and the Glycan Tune products, you can take them beyond a 1x concentration. So in the case of A+ and B+ for both EfficientFeed and Glycan Tune, you can concentrate them anywhere from 1x all the way up to 3x. And then for EfficientFeed C+ and Glycan Tune C+, you can concentrate those products anywhere from 1x all the way up to 2x. And so in what we're calling super concentrating, taking these up to those either 2x or 3x levels, you can gain efficiency with your bioreactor, which I'll show on the next slide. And then you can also get a higher yield. You can get more protein because you're not diluting your culture as much. And then there's also a cumulative effect that over time, if you're using a more concentrated feed, you can get about 10 to 15% more yield just by using a concentrated feed. So what do I mean by maximizing the efficiency of your bioreactor? So if we take, let's say, a traditional bioreactor and this left bar over here, what you have is, let's say that you were going to use a feed at 1x and the protocol required feeding 30% over a length of time. And if you have a 100-liter bioreactor, you know that you just can't add 100 liters of media and feed. You have to account, 100 liters of media and cells. You have to account for how much feed you're going to be adding. So you know you're going to be adding 30% feed. You have to set aside roughly 30 liters of your reactor just for your feed alone. So you're really starting your culture at about 70 liters. If you were going to use, let's say, that same feed, you still have to do 30% feeding, but now you can concentrate that up to a 3x. So what that means now is now you've got a feed that you no longer need to use 30 liters of it. It's 3 times as concentrated, so therefore you only need to you can use 3 times less. So in that case, you're only using 10 liters. So in this middle example, if you were starting your reactor at 71 or 70 liters, and now you've got 10 liters on top of that, that leaves you with a working volume space that's not being utilized of about 20 liters. So what we're saying is when you start your experiment, now you no longer have to start at 70 liters using a 30% feed. You can concentrate the feed, and you can start at a much higher volume to start your experiment. So you're really kind of maximizing that space in that reactor. You're starting at a higher volume. You're adding the same amount of nutrition to your bioreactor, but you don't have to add as much of it. In fact, in this example, 3 times less. I spoke a little bit earlier about the simplicity in reconstitution. And again, this is as simple as adding water. You can see at the top, there's about 6 steps to reconstitute Glycan Tune and the EfficientFeed Plus products. You add water, you mix, you QS, you bring up to your final volume, you mix it just for a little bit longer, you go ahead and filter it, and it's ready to use. You can see with other single-part feeds, there's the opportunity to introduce a lot of error and variability. You might be adding water, adding your powder, mixing, then performing a pH adjust, mixing, then bringing back to neutral after you cellularized particular components. You're mixing again, you're bringing up the final volume, you're mixing again, you're filtering, and then you're ready to use. And for multi-part feeds, like you can see in the bottom, you can add even more steps onto that. So where this brings a lot of error in, there's a potential to overshoot your pHs. And in that case, if you're trying to bring it back down, then you're probably going to bring your osmo out of spec. More steps, more pHing, more additions. You could potentially have reconstitution errors. You can go over volume and dilute your components. There's always the aspect of the extreme pHs. So it's one thing when you're making this on a bench and it's just 1 liter, but let's face it, when you're in a manufacturing environment, we're going to have reactors that are 1,000 liters and up. And when you're at those sizes, you're no longer using milliliter quantities, very small milliliter quantities of acid or base. Now you're using tens of liters, maybe hundreds of liters. Excuse me. And for the technicians that are working with this, that really prevents a safety hazard. We really want to minimize any kind of unsafe practices or hazardous conditions for all of our workers. And at the same time, you want to minimize any extreme pHs with bioreactor control. So certainly if the feed could only go in at something that wasn't neutral, like the Efficient Feed Plus and the Glycan products can go in at, then you're introducing pH variability within your bioreactor. And now your system's got to compensate for that, which is also going to change the osmolarity and other aspects of the culture and could potentially shock the cells or show up later on in both the glycans or productivity or viability. So we certainly offer the Glycan Tuning Efficient Feed Plus products as catalog offerings, but there's a lot of customers out there and a lot of people looking for custom things. And we have the tools to work with you. Certainly we have the ability to customize any of the packaging you might be looking for, so you might want to buy a catalog product, but maybe the packaging isn't what you're looking for. We can certainly work in that regard. On the other side, we can customize the products themselves to meet a customer's needs. We can certainly change, modify those glycosylating and modulating components to add more of or less of. And then we also have a services group that has offerings to help a customer that maybe has a media and feed that they don't want to give up, that they really want to use that, and they don't want to, maybe they don't want to use the Glycan Tune products, but they still want that added advantage of those glycosylation modulating components. So in that case, we have a services group that can work with you, the customer, to develop a media or a feed based on a current formulation that you might have and incorporate these technologies to help target specific glycan profiles. So this is a case study that we had. And in this case, a customer liked their formulation, but they were not getting the product quality that they needed. So the goals were that we had to be able to modify their formulation, but still maintain their titer levels. What good is it if we can get the glycan profile that they have, but now they've got 50% less productivity? And on top of that, they had 4 out of 12 glycan parameters that were matching their target. The remaining 8 they weren't even close. So we had to work to make all of that acceptable for them, maintain that titer, and try to match all those glycans. So we've got a process where we've got multiple rounds of optimization, where we're using, DOE, design of experiments. And we base this on both literature and in-house knowledge from hundreds of these experiments that have been performed. And this was all done through high throughput means. We have amber systems that we can utilize multiple bioreactors at the same time to test lots of conditions. And then we can confirm this with the customer to see if it's meeting their needs. And in this particular case study, it was a success. 9 out of the 12 parameters for glycan targets were hit just about dead on. And the remaining 3 were within 80% of what the customer was desiring. And overall, the customer found this very acceptable, and they were happy to move forward with that customized formulation. So to recap, there's a lot of advantages for using the Efficient P Plus products along with the Glycan Tune products. With Glycan Tune, you're able to target your glycan profiles, showing that transition process that I talked about, where you switch from either a feed of your choice or an Efficient P Plus product to Glycan Tune Plus. I demonstrated that you're able to control your glycans, and you're able to decrease G0F from about 90% down to 45%. And I know I've seen examples where you can get even lower than that. Depending on your setup, you can get down into decreasing G0F down to maybe 30%. And then through working with our customs group and our services group, there's the ability to reduce G0F even more. We know that we can take these components to higher levels if desired. That could, like I said, maybe even reduce G0F down into the 30% range, maybe even lower. And I also showed that using Glycan Tune didn't detrimentally affect growth or product titer. They remained very similar to the control conditions. At the same time, there's also an efficient process for these feeds. Again, it's a simple dial-in process. We wanted to make a product that was easy for a customer to use. There was no guesswork in it. You start with one feed, you transition to the Glycan Tune feed. Or if you're just looking for the best glycosylation results you can, you feed nothing but Glycan Tune. Also showed the ability to reduce the feeding volumes, giving you more flexibility with your bioreactors. In that case, you can what we call super concentrate the feeds. You can bring them up to a 2x or a 3x. And at the same time, you can deliver more nutrients. I talked about that there is a cumulative effect. If you're using the concentrated feeds at a 2x or a 3x, you will see maybe a 10 to 15% benefit over the length of your experiment in regards to product titer. And then if you chose, you could certainly, at a 2x or 3x concentration, maybe add even more of that feed to your bioreactor without having to add a significant amount of volume. And then lastly, I spoke about the reduced risk. So because of the ease of reconstitution, just adding it back to water, mixing, and then filtering and getting ready to use, you minimize all those errors that you can introduce with pHing, multiple pHing steps. You're just adding water, and you're just adding the AGT, and it's ready to use. And then lastly, both with the Efficient P products, Efficient P Plus products, and the Glycan Tune Plus products, we do have some ability to keep these products stable at room temperature for up to 30 days. So we've gotten questions before where a customer wants to attach their feed to a bioreactor and they want to leave it protected from light on the bench. Maybe they don't have the means to refrigerate that, and that's okay. These feeds will remain stable for 30 days. So I'd like to thank everybody that's on the line for attending the webinar, and I'll turn it back to the moderator so that we can take some questions.
Thank you so much, Ryan, for your informative presentation and really important research. We will now start that live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on that green Q&A button at the lower left of the presentation window, type your questions into the box that appears on the screen, and click the send button. We'll answer as many questions as we have time for today. So let's start with our first audience question. How do I choose which Glycan Tune product to pick?
So I guess there's a couple answers for that. One would be if you're already using an Efficient Feed Plus product, an A+, a B+, or a C+, then I would recommend transitioning over to the corresponding Glycan Tune Plus product, the A+, the B+, or the C+. If a customer is using their own media and feed combination, I generally recommend starting with Glycan Tune C+, and seeing where that gets them. Certainly, they can go ahead and test all three of the feeds. It really depends on the cell line, the media, and the process that's involved, but there's a chance that one of those three feeds will match up. If that doesn't work, then certainly we can go the services and custom route, where we can go ahead and work with the customer to try to get that to fit their system as much as possible.
Okay, great. Thank you. I'm not sure if you answered this one within that question, but an audience member would like to know, can they use Glycan Tune with their current feed?
Yep. And I showed an example of that in the presentation, but again, it's certainly a possibility. I've seen it work. There's no reason why that can't be done.
And an audience member would like to know, how do I match a Glycan Tune product with a feed from another vendor?
So it's a little bit of a trial and error basis, but not that much. I would say, I might have sort of answered this in another question, but I would probably start with Glycan Tune C+, and try that with the current feed, and maybe trying that at a similar feed profile, at a similar concentration. Certainly you don't have to go as far as concentrating to a 2x or 3x. These products certainly work at a 1x concentration. And you can certainly use your feed profile that you currently have set up, and then you can do a simplified experiment where you take a couple transition points, excuse me, and transition to, let's say, any one of the Glycan Tune products, and then see where that gets you. And then you'll know if maybe you picked a day, you picked even days for transitioning. You have maybe two conditions, day 10 and day 12. You need a Glycan profile somewhere in between that. Then you narrow down based on that experiment where you could pick a transition on day 11 to kind of really target that specific Glycan profile.
Wonderful, thank you. We have some great questions coming in from our participants. Here's another. What do I do if I want to get less go?
Okay, so that's the G0 peak. So that would be, in this case, what I showed was you can certainly start with just the media and the Efficient Feed Plus products. Because there aren't glycosylation modulating components in either one of those, you'll get a high G0 peak. I know that there are situations where maybe a customer is using a media and feed of their choosing that maybe have components in it that do shift glycans. In that case, that's a little harder to work with. If it's a feed of their own, then certainly that's an opportunity for a services project where we can modify that formulation so that they don't get as much glycosylation. But I think, in the examples that I showed, the only way to get to that higher G0, G0F level would be to not use the Glycan Tune products, or make a transition later in culture, so you're not feeding as much of those glycosylation enhancing components throughout the the run of the experiment. That way you'll see higher G0F.
Great, thank you so much. Here's our last question from our audience members. My media alone pushes to the limit too far. What can I do?
Again, that's a case where, you know, if that's a commercially available media from somebody other than Thermo Fisher, I don't know how much we could do with that. But if it's a custom media or something that a customer is using and they have that formulation, they know what that formulation is, and they're willing to share that formulation with our services group, that we can go ahead and run those DOE experiments and small reactor studies to test what kind of levels of some of these components would get them to the right glycan profile, or kind of back that off a bit so that it's not pushed too far. I think we've got another question that came in too.
I think so too. Here's another one. Can GlycoTune be used in research applications or specifically for bioproduction?
Excuse me. Yeah, it could be used in any situation really. In my case, I'm using it at more of a small scale, flasks, small scale bioreactors, maybe half-liter reactors. And it certainly works in that environment quite well. Again, obviously that can easily scale up to much larger sizes, and I know it works at larger sizes. And I showed examples using CHO DG44 cells, but there's no reason that this couldn't be tried with other cell types. Again, 293 cells maybe, a cell line of your choosing that you want to get a better glycosylation profile. So yes, the product is sold at as low as a 1 liter pack size. So certainly we can, a customer could purchase that and try that at a research scale at whatever size they deem necessary.
Great, thank you. Thank you again, Ryan Boniface, for your presentation. I'd also like to take the opportunity to thank LabRoots and Gibco, part of Thermo Fisher Scientific, for making today's educational webcast possible. Audience members, additional questions will be answered and published on Thermo Fisher Scientific's website. You will receive an email notification when answers are available. Before you go, just a reminder to let everyone know that today's webcast will be available for on-demand viewing through April 2018. You'll receive an email from LabRoots letting you know when that webcast will be available for replay. Please share this announcement with your colleagues who may have missed today's live event. Enjoy the rest of the virtual conference. That's all for now. Thanks for joining us. We'll see you soon. Goodbye.